All right, everyone. Welcome to week 6. We are going to start today moving into content related to research design. We'll spend the next, I think, 2 weeks or so talking about research design approaches. Today we'll be focused primarily on quantitative methods, and the next week, Dr. Christine Borst, one of our other faculty members, will be talking about qualitative methods. Most students historically have utilized quantitative or mixed method approaches to their CPs. It's not to say that qualitative is not possible, but that should be directed based on your research question and whether or not that's the most appropriate way to answer the question that you are asking. As always, before I hand over to Dr. Kessler and Stephanie, just a little bit of grown research / potentially mental health related cartoons. So we have a character saying, it's hard, the way I feel is hard to quantify. And somebody asking, how hard? On a scale of 1 to 10. So we're going to talk a lot about methods that allow us to get at kind of some specific data and numbers versus themes, which is more common with qualitative. And as our general with housekeeping, as a reminder, there is nothing due this week. We ask that you give your instructors, as I've mentioned before, 1 week to return feedback on assignment number 1. Your resubmit due date for it, if you do not earn a 12 out of 15 on this first round, is on February 25th. You're always welcome to resubmit it before that date if you want to kind of get it behind you. Keep in mind that you're only given 3 opportunities to submit, so you get 2 resubmit options for this assignment. And that's really key because you want to make sure that you are taking your instructor's feedback into consideration and thoroughly implementing their feedback when you resubmit, rather than rushing to resubmit, because you don't want to hastily go through implementing their feedback and potentially lose out on a submission opportunity as a result. I did just post an announcement a few minutes ago in the course shell. Your instructors typically give feedback in 3 different ways. One is maybe some narrative feedback in this kind of like chat box area of the course shell that you should be able to see. We all fill out the rubric for the assignment, so you should be able to see exactly which elements of the rubric you may have missed points on. And then most of us, I think all 3 of us actually, will also attach a document to that little text chat area to it. I've heard from a couple students this semester, they've had a hard time. They might be able to upload or download, the file that we attached, but they're not always seeing the track changes. If you're not seeing the track changes, email your instructor and we can email you the Word document again. Blackboard is intermittently weird with attachments in that regard. So some have worked, some have not. It used to be my understanding that it would work if you had it in a certain file format, but not in others, but I've heard from students either way. So if you're having a hard time, if your instructor provided you with an attachment and you don't see any feedback, know that the feedback's there. There's just something wonky going on, so email your instructor for that, and Dr. Del Papalicio and Dr. Murphy are both aware of those concerns as well. You also should be working on your assignment number 2, which is your lit review. We'll talk about this assignment in specifics more next week, but remember, the last time Stephanie was here with you doing a guest lecture, she spent all of the time talking about writing a literature review. So you can have the ability to be working on that assignment now, and you should in fact be working on that assignment now. Utilize all of the resources. I believe it was from week 4 that we covered literature review. So go back to week 4, look at that content, rewatch Stephanie's webinar, and then we will also talk about the specifics of the assignment next week. But without further ado, I'm going to hand it over to Dr. Kessler. And Dr. Kessler, just so you know, I mute my mic and turn off my camera while you're talking as well. But I'm still here if you need anything. So.
Thank you so much. Hi, I'm Roger Kessler, and I'm here with my wonderful colleague Stephanie Brenhofer, and we're going to talk about quantitative research. We're going to talk about thinking about research, things that you should just have as kind of guiding rules, pros and cons of quantitative methods, independent and dependent variables, moving on to reliability and validity and types of study designs. So why pay attention to quantitative research in the general case? Well, one reason to think about doing that is that to help you make your mind up about how you're going to think and behave, to minimize errors in thinking, errors in being. What comes to mind, for example, is over the course of years, there have been many ideas that have been postulated that because of the research enterprise and quantitative research have been dismissed or debunked or in some cases supported. At the moment, we are rigorously trying to look at the idea of integrated care and different models of developing integrated care across 40 sites around the country. And the issue winds up being when we are confronted as clinicians or managers or decision makers with selecting a co-location model as contrasted with a more rigorous integrated care model, can we in fact do research to demonstrate which is more effective? The interesting thing about it is, yeah, we can. We haven't much in our field. Our field has been terribly lacking in its attention to research and evaluation, and hopefully we're changing that, and hopefully the work that you guys are doing will further the field. And if you're going to be at the March conference, we have a number of presentations that should assist with that. So the project that I mentioned is time-consuming. It is 5 years worth of time consuming before we are going to get to the point of being able to establish what we found out and establish the findings. And that's always the case. You're always trying to pursue rigor. You're always trying to generate knowledge, and it's going to take a lot of time. That's always going to be the case. So the primary focus of quantitative research is to answer questions, is to test hypotheses by identifying the relative significance of an answer to numbers, sizes, reactions, and results. Numbers could be things like numbers of inches, or larger or smaller, or more comfortable or more anxious, and generating results of those questions to be able to respond to the questions that you're initially asking. So I was just looking at a new volume of a journal that came out last week or so, 2 weeks ago or so, Clinical Psychology in Medical Settings, in which there's an entire section on the primary care behavioral health consultant model. And part of what reading the introductory paper reinforced for me is that every time you write a paper arguing a position, it's already out of date because the thinking has moved, it's been self-correcting, the prevailing wisdom moves. I'm going to be doing a panel in March asking whether the measure, the PHQ that you're all familiar with, is still relevant to primary care integration, and my position is it depends. That's the answer to every question. Let's see. Yeah, Colleen just made that excellent point that she wrote a paper for that edition well over a year ago. And now it's published, and things have already changed. So the people on this panel are Kurt Kroenke, who invented the PHQ, and John Ware, who invented some other measures, and contemporarily is saying, no, I don't want to measure depression. I want to focus on measuring function, measuring psychological function, measuring physical functioning. So while the PHQ had a significant period of time of being the gold standard measure within primary care, it probably is no longer that, and things have moved. We never prove anything. We just add to the puzzle. That makes sense. Well, glad. If anybody has any questions, please feel free. So these are three core issues that you will consistently come in contact with as you think about, perform, and read about research. We're consistently talking about the empirical verification. So most of you are familiar with a measure that I developed years ago called the practice improvement, the practice integration profile, the PIP. And we decided that we would come up with an empirical way of measuring degree of integration. And we did a pretty good job initially, but now we're in the point, as I just pointed out, we're looking at the measure, we're looking at the items, and we're saying contemporarily, do they still make sense in that same way, or do we need a different version, version 2, to be able to do that? So, parsimony, that is trying to look at something and come up with the way of interpreting it that makes the most sense with the least effort. So somebody wound up saying once, maybe it was H.L. Mencken, that there are 1,000 simple solutions to any problem, and most of them are always wrong. So part of the work of research is to take truistic thinking or common sense thinking and evaluate its accuracy and determine whether simple explanations will affect or we need to look at more complex solutions, which is usually the case. You're all familiar with cause and effect. There are those who argue that the purpose of research is to establish cause. That is, to isolate that particular area, item, measure to explain phenomena. And then lately what's been happening due to the work of people like Russ Glasgow and the ReAIM folks, the question winds up being not so much cause, but what about the effectiveness? We're not looking at simple phenomena. If you look at primary care behavioral health integration, is it a unitary thing? No, actually it's a dance. And it's a group dance of multiple different sets of people doing lots of different things that hopefully will generate an effect that we can then measure. So for me, as a mixed methods that you'll hear more about scientist, cause is an interesting phenomena, but generally on the ground, in the settings that we all practice and work in, we're not looking at unitary dimensions. We're looking at multi-level things that we're doing that in toto may wind up having an observable effect. You're going to see discussion of probability. That is quantitatively assessing whether something is more or less likely. And last, part of the conundrum of research is doing things that are so idiosyncratic that if you want to say, okay, now I'm now going to do that in a different setting, it can't be replicated. An example of the importance of replication, designs that are replicable, is if you folks from your knowledge base were to say the area of the integration world that has the most research support, what would you say? Any idea? Is there a body of research that you could point to that says, you know, here's something that really supports integration? What would you say, if anything? I'm waiting for Ginger. Outcomes. That's great. And the place where outcomes have been looked at the most has been around the issue of the behavioral or psychiatric or care management of depression in primary care. And that work has been done mostly at the University of Washington. And it's generally called the impact model. And the impact model, if you go to the literature, has tons and tons and tons of RCTs, of clinical trials that demonstrate its, randomized clinical trials that demonstrate its effectiveness in practice. In very controlled, specific environments that are able to be managed in that kind of way. So said those folks, well, we can replicate this, and we're going to do that. And they did that in a trial in northern Minnesota, of all places, in a project called the Diamond Project. And the short story is the Diamond Project attempted to replicate the intervention that was the impact work in Washington in on-the-ground practices that did not have the kind of support that other places have. And because of this issue of the importance of replication, let me just sip water for a second. The Diamond Project's finding suggested that the intervention did not work in on-the-ground practices. And I'm sure there will be more trials on top of that. Thus, the importance of replication. One of the dilemmas that we as folks conducting evaluation and research need to pay attention to is the researcher's relationship with study participants. So here's another question for you folks. If we're clinicians in practice with our patients, are there concerns about doing research on that patient group? If so, what are some of those that you might think about? I look forward to seeing this typing. That's great. So the researcher is always going to be concerned, or the evaluator is always going to be concerned about all the factors, including the relationship with the patient, affect study results. For example, if a funder says you need to demonstrate that what you're doing is effective, otherwise we're not going to fund you anymore. Boy, that is something that could impact on the results of what you do. Or, for example, if you're going to say, well, I'm going to look at patients in this clinic, and I'm going to apply statistical analyses after a particular intervention. Well, the analyses that you select and how you think about the sample in terms of normal distribution is going to affect the measures that you use and the results that you wind up getting. So, as Dan noted, that if you're asking patients for data, then there may be a bias towards the patients wanting to please the clinician. Christina suggests that that kind of work could sometimes be tainted. And being familiar with your subjects may sway your interpretations. All great points. All great points. And all the things that we need to pay attention to. On the other hand, I don't know if this has happened to any of you folks. There are some research teams that are so concerned about this, they'll bring independent data collectors who don't know anybody into, for example, an office or an office suite or a waiting room and collect data in that way in ways that are very obtrusive and often not respecting the culture and flow of the practice. So I understand there was a Super Bowl a couple of weeks ago. Since I'm originally from New York, the idea of a Super Bowl between, I think they were called the Patriots and the Eagles, is just a phenomenon that can't exist and you can't pay any attention to. So, but whatever, I had football on my mind. So I had this imagery of when does the penalty flag get thrown? Sloppy thinking, that is your theory and your construction and your design are not well thought through. Not doing sufficient planning. One of the things that we hear in the program often is, well, I'll just get that data out of our EHR. And then a short period of time prior to the data being due, there is the discovery that either there were processes that needed to be gone through, or there was just an inability to generate the time frame in that period of time. Carelessness. There's always a focus on rechecking data collection, storing it in a particular location, and being very clear how any of the data winds up being entered. And unacknowledged bias, which means that you're participating with biases that you may not be aware of. So part of the work in your CP is to think through what are you bringing to the investigation that you're going to do that might potentially compromise some of the work, and clearly acknowledging those and how they might influence your work. So I would suggest that when you discuss your projects with your CP chairs, one of the things that you spend some time with is looking at whether there are, in fact, any unacknowledged biases that you need to pay attention to. So when we think about randomized control trials, we think about trials that are internally valid. That is, we have removed as many different possible sources of contamination as possible, such that the result probably is a base reflection of the phenomena being tested. The conundrum is the other side of internal validity is external validity. So what's the percentage of behavioral randomized control data that is generated and makes its way into practice, into clinical practice of behavioral health or behavioral health integration? And how long do you think it takes for that to happen? Any thoughts? So one of the great thinkers in our field is a physician from San Francisco whose name is Larry Green. And Larry Green is the one who has popularized what I'm about to say. And Sean suggests less than 5% and greater than 2 years. You're a very cynical man, Sean. Or woman, I'm not sure. but you're darn close to being accurate, the data appears to suggest that about 17% of RCT research makes its way into clinical practice over the course of 17 years. So not much ever makes it into practice, and what does make it into practice takes a long time. And part of the problem of it taking a long time is there is little attention to context. There's little attention to the things that are important to the community, to a practice. There is little attention to how a particular piece of research affects the flow. Often patient populations in trials don't resemble the patient populations that we see in practice. So given all that, there has been this evolution, as Colleen was mentioning. Next week we'll talk about qualitative research. There's an organization in NIH called the Office of Behavioral Science Research, OBSSR, and they have published a report a bunch of years ago now on what they call mixed methods research. That is the integration of quantitative and qualitative research to both look at the rigorous issues related to the internal validity as well as being able to consider the external validity issues to come up with methods that can describe the both. So this is generally a quick way of thinking about the pros and cons of quantitative research. One can generate clear interpretations. One can organize thinking and perception, generate logical, sequential, controlled experiments, limited researcher bias, and they should be able to be more broadly understood. On the other hand, it can't help understanding issues that haven't been identified as a focus of the study. I'm working on a disparities project at the moment. And one of the questions that we're asking that hasn't been asked is there a difference in the healthcare that is provided to communities that tend to be heterogeneous as opposed to the healthcare that's provided to communities at risk for disparities that are more homogeneous? Now, let's just assume for a moment that there is that effect, that there is that difference. If we didn't think to test that difference, then we wouldn't know that that's one of the underlying factors affecting the research. You're limited in the amount that you can study at once in a quantitative design. And as I was talking about, if you look at integration and say, okay, I'm going to do a quantitative study on integration, I would say, fine, define for me the intervention that you're calling integration. And I would bet dollars to donuts that you would list off multiple complex elements that are contributing to this thing that's integration. Evidence-based treatments, communication with physicians, warm handoffs, using the same physical space, whatever it winds up being. And in quantitative research, it's more and more difficult to be able to tease those out. So here is a very rough framed notion of a hypothesis. It's an idea that you are going to test through systematic investigation. The issue of evaluating something in a way that is not through systematic investigation has led some to say you're not really doing hypothesis testing, you're just doing idea generation and looking at what might be later hypotheses. So that's an issue that will come up time and time again. A hypothesis is a prediction. It's you saying that intervention X will find no difference compared to the control group, thus what's called the null hypothesis. And hypotheses are most frequently discussed and generated in experimental research, in which, as I said, it's mentioned as hypothesis testing. So getting into the weeds a bit, you are going to try 4 weeks of treatment X. So an independent variable is the amount of time that is controlled or manipulated by the researcher. And you're saying that intervention over that period of time is going to have some effect on the patient. The patient's response can be worse, it can be better, it can be the same, some effect on the dependent variable. And you can think of it as the difference between the treatment and control group. The one thing that makes them distinct. The dependent variable on the other side of that equation is that which you are measuring for difference, or calling it the outcome. That's the thing that is influenced by the intervention you're doing. So does motivational interviewing support patients being more active in their treatment? So in that question, what's the independent and what's the dependent variable? Anybody have any idea? Motivational interviewing affects patients engaged in their own care. Which is independent or which is dependent? Great, great, exactly right. Exactly right. And it can get tricky sometimes, but always think through the issue of identifying. You're going to be asked when you develop your plan for your project to identify the dependent and the independent variables. The struggle for us, I'm wondering whether I'm starting to get into the material that you're talking about, Stephanie, and just give me some quick feedback about that. Where has most of the research been done? Most of the research has been done, behavioral research has been done, replicating drug trials, that is, using RCT models focused on internal validity, and that amount of work far exceeds external validity. And there are just some citations that support that. Here's a way of thinking about reliability, validity, or both. Let me just see here. I'm not seeing anything from Stephanie, so I'm assuming that I'm getting very close to when she's going to take over. So, aha, it's my last slide. Well, I'll take about 20 minutes on this slide then. No, I'm just about done. Part of what you're going to have to grapple with, and we grapple with this all the time as researchers, is, yeah, you can collect numbers, but those numbers need to be reliable and valid. And there's only one of these targets that's an example of reliable and valid data. And obviously, it's number 4. So with that, yeah. With that, Stephanie Brenhofer is going to take over. I'm not hearing.
Turn on your mic.
Sorry, I turned my mic off. It is on now. Thanks for that, Roger and Colleen. So my section, we're going to be talking about types of study design. And there's many different types of study design. We'll just focus on 5 that I have listed here. So we'll go over what cross-sectional...
Hold on, I'm going to interrupt you real quick, Stephanie, if that's okay. I'm having a hard time hearing you, but I think it's in part because Roger's mic is still on as well. So Roger, if you can
turn my mic off,
then Stephanie, just make sure to stay close to your internal microphone so we can make sure we still hear you.
Can you hear me better now? Or is it still bad?
I can. Yeah, it's better.
Okay. All right. So for my portion, we'll go over different types of study designs. And while there's many different types, I'll just go over 4 study designs. And really the last one, the meta-analysis, is just a little bit different. And you'll surely find out why. So cross-sectional studies are basically a snapshot in time. And what I mean by that is that you're really just taking one time point in a person's life, and you're trying to understand a lot about that one time point. And because it's just one time point, we can't infer causality. So we really don't know what is influencing or what is causing what. We really only know what data we gathered at that specific time point. And that's why the language that we use to describe these types of studies, and you'll see this in the literature, is that they always refer to what they're looking at as associations or relationships. Because we can't infer causality because we really don't have enough data to figure out if X is causing Y or if they're merely just associated with each other. So some examples that can be made if you have a cross-sectional study is that someone's hemoglobin A1C impacts levels of depression. And what we cannot say is that hemoglobin A1C influences levels of depression. To use the stronger language of influence, we would need to conduct an experimental study to show cause and effect, something that we can't do with a cross-sectional study. So this is just an example of what a cross-sectional study looks like. So for each of the different types of study designs, I give you an example of what this could look like. So I grabbed one of Roger's papers. And the important thing to note with these examples is that I specifically sought out good examples for each of the study designs, but it's not always clear when you're reading the literature what study design, different types of research fall under. A lot of the times they seem to pull different aspects from multiple different designs. So it's not always clear, even after you read the whole paper, let alone from just pulling from the abstract. But if you look at the abstract for Dr. Kessler's paper, you can see in the method section where it's very clear that they did a cross-sectional study. And that they just surveyed people at one point in time, and then they used their responses from the survey to inform their hypotheses. So the next type of study design that we have are case control studies. And if any of these don't make sense, feel free to raise your hand or write a comment and I can try to clarify anything for you. So with a case control study, we're looking at an observational retrospective design. So what I mean by that is that the exposure and the outcome have already occurred. So you're specifically seeking out people that have a specific disease or a specific outcome, and they're your cases. And then you're specifically seeking out another group of people who do not have the disease or the outcome, and they're your controls. And you're going to compare these people that have the disease and they don't have the disease, and you're going to look back on their exposures. So you're trying to figure out what previous exposures could have led to this specific outcome. So just an example is if we took people that have lung cancer and then we took people that don't have lung cancer. So if you have lung cancer, you're the case. If you don't have lung cancer, you're the control. And then we may look back to see if they were a smoker or a non-smoker. And then that's really what the exposure is. So, you know, were you exposed to being a smoker or were you not exposed to being a smoker? And the unique thing about case control studies is you can actually study multiple exposures. So you're not limited to just one, but for simplicity purposes, I only listed the one. So the other thing with case control studies is that we're going to match our subjects. So I put that half of the subjects have a certain outcome and half don't. And I don't mean that in like a 50-50 sense. What I mean is that you're going to have cases and you're going to have controls, but they're not in equal proportions. So in general, we have 2 to 4 controls for every one of our cases, and we match them on similar characteristics, and we do this to try to limit any type of bias or confounding. So we want our subjects in both groups to be as similar as we can. So usually they're matched on 2 to 4 characteristics, which might be age, sex, potentially race, socioeconomic status, anything that the researchers feel is most important given the outcome that they are studying. And what's also important is that the cases and the controls come from the same population. And I don't mean like the same geographic population or the same like racial or ethnic population. What I mean by that is where you're actually getting your participants. So if you're getting all of your cases from the local hospital and you're getting all of your controls from the local like fitness center, that's a problem because now they're not from the same population and the outcomes could be drastically different because of simply where you found your patients, or your participants. So what's more appropriate is, let's go back to the lung cancer example. If we're finding all of our patients diagnosed with lung cancer at the local hospital who are like currently in the hospital or even at a clinic, we also want to pull our controls from the same location. They're just maybe in the hospital because they have the flu or they broke their leg, anything that's not related to having lung cancer. And that would be a more appropriate way of getting your participants, because if you don't get them from the same population, you're introducing biases into your study. Another important factor is that all of the cases should be recently diagnosed with the illness. And the reason being, and Dr. Kessler sort of touched on this before, is participants needing to recall their past. So with these types of studies, we often ask people about their previous exposure. You know, how many packs a day of cigarettes have you smoked for the last 10 years? What was your fruit and vegetable consumption? How physically active were you? And we want to capture all of this before someone's diagnosed, so it's really important that they're recently diagnosed, because if we let somebody enroll in our study and they've had the disease for 4 years, they're going to have a really hard time discerning what they had or what they did before they were diagnosed with the disease and then what they've been doing the past 4 years. So that just helps get us a clearer picture as to what happened before they were diagnosed. So this is an example of a case control study, and these authors were really nice, and they told you in the title that it's a case control study. But you can also read the abstract and see what their control group consisted of, along with their case group as well. So next we'll talk about cohort studies, and there's two different kinds. So there's prospective and there's retrospective cohort studies, and I sort of made this figure to help differentiate between the two. So prospective studies, we have our study initiated in the middle, and with a prospective study, we assume that they've already been exposed or they haven't been exposed to whatever it is that could lead to our particular outcome. We enroll them in the study, and then we follow them up over a period of months or years, decades maybe, and then we see who gets the outcome and who doesn't get the outcome. And we're looking at that in both the exposed and the unexposed groups. But with a retrospective study, everything has already occurred in the past. So when we start our study, our outcome of interest and the exposure have already happened. And now we're looking back to see when that occurred. And that sounds a lot like the case control study, but in the next slide or two, I'll clarify how they're different. So we'll start with the prospective study, and this is an observational study design. And you generally start with a healthy population, but really they don't need to be healthy in maybe the strict sense that we would think of people as healthy. They just won't possess the outcomes that we're interested in. So they might not be physically active, they may be smokers, maybe they have diabetes, but if we're not interested really in any of those things, then they'd still fall under like the healthy population. They don't have what it is that we're interested in studying. And really you just measure them over time, and you see who develops the disease and who doesn't develop the disease. Some cohort studies get real fancy, and I won't get in depth with this, but you can actually nest a case control study within a cohort study, and that sort of adds to the rigor of that study design, because as people are diagnosed with a certain outcome, they can become your cases. You pull other people that haven't been diagnosed, they become your controls. So now you have this sub-study going on in a larger study. And this is an example of a prospective cohort study, and you can see that they were followed over a significant period of time to see who developed the outcome of interest and who did not. So for retrospective cohorts, your outcome has already occurred. So like a regular prospective cohort, we're not following people over time to see who gets the disease because they already have it. But how this differs from a case control study, in where we're first seeking out the diseased population and looking at their exposures, we do the opposite with a retrospective cohort, and we're first identifying patients based on their exposure, and then we're comparing their outcome. So in a case control, we took people with lung cancer, people without lung cancer, and then we looked back to see if they were a smoker or non-smoker. In a retrospective cohort, we're taking the smokers and the non-smokers first, and then we're examining to see if they developed lung cancer. So oftentimes, what'll tip you off that it's a retrospective cohort, because they'll still gather data over a period of many years, it's that they didn't actually enroll participants in their study, but oftentimes they use medical records, or they use like cancer registries or other national registries to gather this information. Okay. So this is an example of a retrospective cohort study, and using medical records. Okay. So on to experimental studies. Oftentimes, these are referred to as randomized control trials. And in this type of experiment, or in this type of study, participants are randomly assigned to either receive the intervention or the control. And then they're followed over a certain period of time to see if the intervention and the control group differ. So based on the intervention, at the end of the study, is the outcome different? Did one group fare better than the other, or are they equal? So in this type of study design, it's highly controlled to the point where the researcher is manipulating nearly every part of the study. And so for example, if we have, like let's say we're doing a study to see if physical activity would decrease patients with diabetes, their hemoglobin A1C. So our intervention group might have to exercise for 30 minutes a day, 3 times a week for 6 weeks. And then our control group receives a pamphlet on physical activity. So both groups would get their blood sugar tested at the beginning of the study, chances are multiple times during the study, and then at the very end of the study, and then we would compare and see was there a significant decrease in somebody's hemoglobin A1C. And actually it would probably be much longer than 6 weeks because hemoglobin A1C is really reflective over the previous 3 months. So participants in this study might be enrolled for quite a while to try to capture a meaningful difference. But that's just one example of people being randomly assigned to a group and then told what to do and then measuring them at follow-up. And then this is the one type of study design where we can really say, A causes B, and we get to use words like the effects of or the influence of, because we're pretty certain as to what is going on. And this is just an example of a randomized control trial, and they actually did like 1 year follow-up. So quite a bit of time checking up on the patients or the participants. So for our last type of study design, we'll go over meta-analysis. And I just want to clarify that a meta-analysis is not synonymous with systematic review. So systematic reviews are when you are sort of systematically taking the literature. So you're taking pretty much everything that's been written on a certain topic and you're synthesizing it into one paper, and you're really telling everyone what has already been done in a specific area. So it's qualitative in nature and there's no additional statistics performed. But a meta-analysis takes the systematic review and adds another layer, and where they are going to perform additional analyses to sort of report in a quantitative way what has already been done and the effects that were seen in other types of studies. So for this meta-analysis, it's really important, and this isn't as important in a systematic review, but in a meta-analysis, we want these studies to be similar enough in terms of their independent and dependent variables, because they're going to be statistically combined and then analyzed, and then we're going to report on whatever it is we're looking at. So if we're interested in diabetes and depression, all the papers that we pull, we want them to be similar in study design, and the independent and dependent variables, pull those findings together and then say, okay, based on 12 different studies, this is the association between these two variables with this specific impact on an individual. So that's really the goal of a meta-analysis, is to bring together the work of other researchers and then say something about it. And it also can distinguish trends, and the effect sizes are super important. And the effect size is just the influence that your independent variable has on your dependent variable, and that can get compared across the different studies that you're looking at, because they'll each have their own effect size for the exposure and the outcome, and you're really combining and bringing it all together. And this is an example of what a meta-analysis paper looks like. You'll see that they search a bunch of different databases and that they statistically combine the results, and that's really the key, that it's a meta-analysis if they don't give it away in the title for you. So we only have 5 minutes left. Roger, do you want to go over this, or do you want to skip to the summary slide and sort of wrap it up?
I'm back. So what I would ask that you do is, between now and next week, review the designs that have just been discussed, identify a research question that's interesting to you, and then try to select which design you might use to study it. And discuss that, and maybe that can be in a discussion board. Reminding you to discuss cross-sectional cases.
Sorry, guys. Just as for all students, this process that Roger is encouraging you to go through, thinking through your research question and what design you might use, is actually part of an upcoming assignment for you. So keep in mind that this process is not moot, and it may not be a discussion board, but you will need to be able to identify your specific research design and then extrapolate on it pretty significantly in terms of what kinds of things you will do to follow this research design for your methods, which comes up in a few weeks. So assignment number 4, I believe it is, falls into this pretty closely. So make sure that you understand this content.
So just going through this little exercise that we're suggesting will put you ahead of the game when it comes around to being a task that you really need to engage in.
Absolutely.
So some summary points. If you have an idea that you're going to get everything else done and then you'll look at your culminating project and find a little time to do that, that strategy is going to suffer. This process will take time and attention, quite probably more than you anticipate. Respect the science, pay attention to the elements that Stephanie and I have laid out, and recognize that, part of the reason that we just don't hand you a diploma before you do a CP is that it's a challenging process. And part of going through it will make you better thinkers, better consumers of the literature, and better practitioners. There are no shortcuts to this. You may try to get away with shortcuts, but hopefully your team and your faculty supports will catch that and point it out to you and push you to eliminate shortcuts and do this in a more rigorous way. That said, define a project within the limits of what you have available for time and what data that's really available and the findings that are realistic for you to generate. I'm going to prove that integration is effective is not going to work because the task is too big, you don't have the sufficient data, and it's a huge amount of time. The bottom line is, whatever your career path, facility with the stuff that we've talked about today will serve you and help you move forward. We have time for a couple of questions if people have any.
I am fine hanging out so that if people have questions, they can ask them. But I'm also cognizant of time, so if people need to sign off, you're welcome to do so as well. But if people have questions, ask away. Take advantage of Dr. Kessler and Stephanie's expertise by all means.
So I don't see any typing, so maybe the best idea is...
Typing. Don't think you're stopping.
Okay.
There
I'll see you
wasn't.
again. Okay, I'm waiting.
Oh, and now it's not anymore. All right. Well, I guess if there aren't any questions, we can wrap up for the evening. Know also that Dr. Kessler is part of our team and a great resource for students if you want to outreach at other points in time. And otherwise, I hope everyone has a good night, and we will see you all next week. Take care, everyone.
Thanks, everybody.